Good afternoon, everyone, and welcome to another great session here at the 2020 Jefferies Virtual London Healthcare Conference. As we were just mentioning, I made it to London. As you can see my background, Bruce and Mark are probably in Boston. I would love to turn the stage over to the team at Morphic. Today we've got the Chief Scientific Officer of Morphic, Bruce Rogers, as well as the CFO and Chief Operating Officer, Mark Sheeran. Morphic has had an exciting 2020, advancing through the pipeline. Importantly, I'm sure we'll talk about the phase 1 with their oral ulcerative colitis program. And without further ado, I'll turn it over to Bruce.
Thanks, Michael. Much appreciated. Good afternoon, everybody. I think we can get started. So thanks all for joining, and thanks in particular to Michael and Jefferies for the invitation. It's much appreciated. It's my pleasure to be introducing Morphic today, where our main focus is leveraging a uniquely deep understanding of integrin biology to discover and develop a wide suite of oral drugs that modulate these very central biologically relevant pathways to combat a host of conditions that unfortunately remain very poorly treated or untreated today. I'm Mark Sadrian, COO and CFO of Morphic, and I'll walk through a quick summary of the company and the key pipeline programs, and then turn it over to Bruce Rogers, our Chief Scientific Officer, to walk you through our most advanced programs in a little bit more detail. Before I start on the next page, I should say that I'll be making some forward-looking statements today. Please take a look at slide 2 here, and also our financial statements filed with the SEC for additional information on the risks of investing in our business. So starting on the next page, on page 3, Morphic is really at a key inflection point right now, and very well positioned for growth into 2021. Our MINT platform continues to deliver highly specific small molecule inhibitors against very validated targets in autoimmune, fibrotic, and even oncology indications. And our lead asset, morf 057, which was designed to replicate the action of Entyvio in its simplest form, but in pill form, has entered the clinic, and volunteers are already being dosed. And our partnerships with marquee pharmas continue to advance very productively. And lastly, our strong capital position on the last slide, allows us to fully fund both of our clinical as well as our preclinical initiatives as we advance into the clinic. Now we can go to the next slide. Thanks. So let me take a minute on slide 4 just to describe the integrin receptor itself and our overall approach. So we often say that Morphic was 30 years in the making, stemming from the original discovery of the class by the Springer lab at Harvard. This is a very diverse and central group of cell surface receptors that signal bidirectionally and are implicated in a variety of disease states. Many biologic therapies have been approved which target this class. But so far, no one has been able to design an oral small molecule which does really the same thing. And the discovery or realization, again from the Springer lab, on how to achieve this ultimately gave rise to our MINT platform and the company as a whole about 5 years ago. Next slide, please. On this page, you can see a brief description of our MINT platform. So I touched on it on the prior slide, but it's really composed of 3 main pillars. So there's a tunable product design engine associated with our long-standing relationship with Schrödinger, in-house biology and translational capabilities, and proprietary expertise. This last point relates to our only of its kind in-house crystal structures and conformational understanding that allows us to deliberately target specific integrins in the conformations that we want to target them in. Next slide. So here on page 6, you can see our key pipeline programs, led, of course, by morf 057, an orally available alpha-4 beta-7 antagonist being explored initially in ulcerative colitis. This program entered the clinic this summer, and we plan to release the top-line SAD data in Q1 and the totality of the phase 1 data by mid-next year at an appropriate peer-reviewed scientific forum. We're also progressing additional highly specific small molecules with potential utility in a variety of areas spanning multiple fibrotic indications and solid tumors. On the bottom half of the page here, you can see our partnered pipeline. And you remember that this summer, AbbVie exercised its option to assume the development of our alpha-V beta-6 programs in IPF and other fibrotic diseases, and AbbVie will be responsible for all the clinical and regulatory updates going forward for these programs. We're also investigating a number of undisclosed targets in partnership with Janssen. And now with that, let me turn it over to Bruce to discuss our key programs and upcoming milestones in more depth, especially morf 057. Bruce?
Thanks, Mark. I'm Bruce Rogers. Thanks for the invite today, Michael, to give a talk on our programs at Morphic. I'm the Chief Scientific Officer at Morphic and have been for the past 5 years. I'm excited to talk about these programs today because it really speaks to what we've been able to accomplish with this platform approach that Mark introduced us to. And if we advance to the next slide, I'll give you a high-level overview of what I want to cover today with alpha-4 beta-7. We've identified a compound, morf 057, that's highly selective based on our platform, and I'll talk in detail about that. Mechanistically, this is a well-understood mechanism of action that has an approved therapeutic in an antibody in ulcerative colitis and Crohn's disease, so the indications are also of significant interest from the patient perspective. We've published over the last year at a number of conferences a really nice package of supporting information from the non-clinical perspective to be able to support going to the clinic. And we announced that we've started this phase 1 study very recently. And I'll go through each of these aspects over the next couple of slides. On the next slide, I want to just highlight the mechanism here because I think it feeds in very nicely into what we're going to talk about relative to the non-clinical characterization. This is a well-known mechanism of the approved antibody Entyvio. I'll often refer to it as vedolizumab, its generic name, but it's an inhibitor of an integrin, alpha-4 beta-7, which is expressed on the surface of T cells. And you can see from this cartoon on the right-hand side, the T cells are in green, flowing through the vasculature space or the blood. And on the surface of those T cells, where you see this punch out here from the surface at a small to a larger level, you can see alpha-4 beta-7 is expressed on the surface of those cells. The ligand that alpha-4 beta-7 binds to is called MAdCAM-1. And it's expressed on endothelial cells in the subendothelial space in the gut. And so it's a highly selective approach for the homing or passing of T cells from the vasculature space to the gut. And what we're looking to do with this, as has been established by Entyvio, is inhibit that process, which leads to a well-established benefit in the treatment of UC and Crohn's disease. This is a significant area, and our approach is moving from an antibody approach dosed in an IV format to an oral pill format dosed with patients on a daily basis. If we move into the next slide, I show you the characteristics of morf 057. This also exemplifies the challenges that we had in terms of identifying a compound within this space. Alpha-4 beta-7 is a heterodimer, as I pointed out, with an integrin that binds to MAdCAM-1. There is a similar integrin, alpha-4 beta-1, which is also expressed on leukocytes, whose cognate ligand is VCAM-1. Now, this is an important understanding because, as you can imagine, half of the binding site for this, where our molecules bind, is identical in that alpha-4 is a common subunit between these two, this heterodimer. The reason we want to eliminate alpha-4 beta-1 activity is because if you know of an agent called Tysabri, this is the mechanism by which that agent in the treatment of multiple sclerosis works. This is an integrin that is responsible for immune surveillance, much like alpha-4 beta-7 is, but it's responsible for immune surveillance to the brain. And if you inhibit this integrin, you risk not having that immune surveillance in the brain, and therefore you have this risk of PML. So our first challenge with morf 057 was to identify a highly potent and selective agent versus alpha-4 beta-1. And I show you that in the right-hand slide of this table here. In table item 1 is morf 057. It's a single-digit nanomolar compound with over 3,000-fold selectivity versus this target, alpha-4 beta-1. There's also one other related integrin, which has a common beta-7 pair, and that's the alpha-E beta-7, which is responsible for attention of T cells in their spaces. And we also have high selectivity versus that for our small molecule. We've characterized this versus a number of tools, vedolizumab, natalizumab, etrolizumab as the antibodies, as well as a non-selective small molecule AGM-300, which is known to show an effect as well in a phase 2 clinical trial. So we've taken our platform, developed this highly selective, highly potent molecule by maintaining it in the oral drug space as well, and we've now characterized it in a number of in vivo models. On the next slide, what I show you is a mouse model here. And what we do is we dose our drug in this mouse model, and it's a mouse model of homing of T cells. So if you think back a couple of slides ago to where we are monitoring the homing of T cells to very specific tissue types, we're monitoring exactly that process here in the mouse. So we dose our drug, and we follow the drug by labeled beta-7 highly expressing T cells that also contain a dye. So we can essentially cell sort and understand where exactly these T cells are homing to. And then we do a FACS analysis versus a variety of tissues. And what I'm showing you on the right-hand side of this slide is a gut-centric tissue, the mesenteric lymph node. What I'm showing you here is the effect of a vehicle, an antibody, and our small molecules. So let me focus on the vehicle bar first. That's the first bar. What happens when we inject these T cells is that about 20% of these T cells actually home to this specific tissue area in the gut. And you can see that in the first green bar on this slide. The second green bar is an antibody called DATK32. We can't use Entyvio in a mouse model because it doesn't bind to the mouse version of this receptor, but DATK32 is the surrogate to that. So we're doing essentially a direct comparison to the antibody here. So the antibody essentially inhibits all of this trafficking of these T cells to this specific tissue. And then we look at a dose response fashion with morf057 from 0.3 milligrams per kilogram all the way out to 30. And we see this beautiful dose response curve where we see these very significant statistical reductions in this homing of T cells. So this gives us a confirmation in a mouse model that we're inhibiting exactly the mechanism that we set out to do. We've now taken this one step further into non-human primate, and I'll show you a couple of slides of data here. The first thing we do is we emulate virtually what we did in the mouse model, but rather than harvesting tissues, we've now developed an ability to look at whole blood, and we isolate whole blood from non-human primates. So non-human primates have circulating T memory cells that express alpha-4 beta-7, and so what we do is we isolate blood. And in the blood, you would expect an increase in the number of T cells. If they're not trafficking to the gut, you'd see an increase in the bloodstream. And that's exactly what we see with this tool compound, MR5288. This is a very close analog of morf057. When we dose it at 30 and 50 milligrams per kilogram, we see about a 1.4 to 1.5-fold increase in these T cells within this population in the non-human primate. Very close to what you see with the antibody, vedolizumab, when it's dosed, and that's a published study that you can compare that to. On the right-hand side of this slide, I'm showing you this activity with morf057. And there's a couple of really pronounced things I want to make sure we point out with morf057. First of all, we do see this 1.4 to 1.5-fold change as demonstrated by the blue line on this graph on the right versus the red line, all statistically significant increases in these T memory cells across the time course of this assay. And the time course we're looking at is a 5-day time course with this assay. But I also want to draw your attention to the fact that these compounds are now dosed orally via gavage in a non-human primate in a BID fashion over the course of 5 days. And we see this sustained ability to change these T cell trafficking counts within this non-human primate model. So really nice proof of concept that we can now not only modulate the mechanism, but we can do it with an oral treatment with our molecules in a low dose fashion relative to what we're seeing with morf057. On the next slide, I bring your attention to a receptor occupancy assay that we have developed, which really starts to now parse out what we can measure very reliably in human and understand just what percentage of these receptors we're fully occupying in the context of this assay. So you can imagine dosing a drug in a human being and drawing blood from that patient. And you have on the surface, as shown in yellow on the left-hand side of this, alpha-4 beta-7 as a receptor. And if you dose then your fluorescent probe, once you isolate that system and you have no drug on board, you'll see a high fluorescence relative to this signal versus full occupancy of the receptor. You won't see a signal with this in this system. And again, we've used this tool molecule 5288 to validate this. And if you look on the right-hand side of this slide, what you see is the lower dose, the 30 milligram per kilogram dose, looking at receptor occupancy. And I'm showing you several time points here. The 2-hour time point, which is the first point that I'm showing you on this slide, completely saturates, as you would expect, the receptor occupancy. So you have 100% receptor occupancy. But then I'm also showing you a 24, a 48, 144, and 168-hour time point. And those time points, we're actually measuring the receptor occupancy just before we give the next oral dose of the drug. And you can see that we're approaching 100% receptor occupancy at virtually all of these dosing paradigms with this compound MR5288. And I don't have it in the slide set today, but we've actually disclosed very recently at UEG the human data affiliated with morf 057. What I do want to turn our attention to is that this sets us up beautifully now to go to the clinic. And on slide 14, I'm showing you what we're doing in the clinic. So we announced a couple of months ago that we have dosed our first healthy volunteer in our SAD study. And that cohort here is ongoing. We're going to be running 5 cohorts within the phase 1 study of the SAD phase of this to be able to build an understanding first and foremost about the safety, the tolerability of these molecules. But as I just set up from the previous 2 slides I've gone through, we're going to look at receptor occupancy as a key pharmacodynamic marker that we're affecting this target. And we'll look at alpha-4 beta-7 as well as alpha-4 beta-1 at trough levels in these studies as we look at them. We're also going to do a small food effect study within the context of our phase 1, as well as begin the study in the multiple dose fashion of this as well. So really important understanding that we're building now in phase 1. As Mark articulated earlier on our pipeline slide, we anticipate some of the top line results for this early next year, and by mid-year disclosing the full cohort of this study in phase 1 at an appropriate scientific meeting. That will then pivot us directly into a well-controlled adaptive trial. We're defining that trial right now in terms of what we want to accomplish in that study, but we'll start in UC patients is what our plan is there. And that will start as soon as we can after the phase 1 study is completed, the analysis is complete, and we've gone back and had the appropriate conversations with regulatory agencies. So we're very excited about this program. It's our first clinical stage program that's a wholly owned program within Morphic, and it really establishes the value of our portfolio, the value of our platform in the ability to establish a portfolio. I did want to just pivot for a couple of slides now into our earlier portfolio. Mark set up this slide very nicely in that we have a platform at Morphic. We're not just going after alpha-4 beta-7, but we're actually wanting to interrogate much of this integrin space. And I have 2 slides here which introduce 2 new programs into our portfolio. The first one of these is an alpha-V beta-1 inhibitor program. This is a highly selective program for only alpha-V beta-1, something we've only been able to do because of our crystal structures of alpha-V beta-1 and related integrin targets. Along the way of executing in our AbbVie partnership programs with alpha-V beta-6, we identified some very highly selective alpha-V beta-1 agents. And what it's allowed us to do is build a much deeper understanding of what the biology is behind these specific integrins. There's a lot of information published out there that suggests that this is a TGF-beta based mechanism. What we know is that there is a reduction in collagen, and we've been able to validate that within the context of our experiments. But over the next year, you could look for a few key publications out of Morphic where we're going to start to tell this story about the biology that's only been enabled by us identifying highly selective alpha-V beta-1 inhibitors. So this is an introduction to this target. We would anticipate the identification of a development candidate sometime in 2021 for this program, and this will be a next frontier of programs within our portfolio. On the next slide, I show you just one slide from our exciting alpha V beta 8 small molecule inhibitor program. What we're looking at here is we're looking at combining this important biology. So alpha V beta 8 is important in the activation of TGF-beta. It's a key regulator, which is a key regulator in tumor formation and metastasis. And essentially what we're looking at here is we're looking at a well-known model, the EMT6 murine breast cancer model, which is known to be insensitive to anti-PD-1 therapies. And I show you on the left-hand side of this slide the data from an anti-PD-1 antibody. And it looks identical to vehicle in this assay. You don't see a response at all. We don't really also see a response with alpha V beta 8 alone as a small molecule inhibitor. But when we dose these two agents together, you can see this bold orange bar at the bottom of this slide. We see a really profound effect on tumor volume within the context of this EMT6 model. And if you shift your attention to the right-hand side of this slide and look at the Kaplan-Meier plot on the right-hand side, you can see that there's a profound impact on survival. That's the orange bar across the top where we see really improved survival versus these monotherapies as we do it. So a good way to think about this is an opportunity to take a cold tumor that's not responsive to PD-1 and make it hot and turn on the system and really provide a pronounced response. We've also introduced this program into our portfolio. It's tracking slightly behind the alpha V beta 1 program, but it's a really interesting foray into an important element of biology around TGF-beta that brings us now into the oncology area as a company as well. So that's a nutshell of a couple of exciting programs that are coming forward within our portfolio. And I think I'll turn it back to Mark now to take it from here.
Thanks, Bruce. So on the next page, you can just see a quick summary of our platform validating partnership. So as I mentioned, AbbVie recently opted into the alpha V beta 6 programs, which carried with it a $20 million opt-in payment. And we'll be due royalties and milestones on further progress there. But AbbVie will be responsible for all of the key updates, regulatory and otherwise, going forward. We're also working with the cardiorenal division of Janssen on multiple undisclosed targets, and those efforts continue to progress on track. So we're very happy with the progress with both of these validating partnerships. On the next slide, you just see our experienced management team and a really great board. So in summary, we're really excited about what's coming down the pike in 2021. Of course, it's been a great year for us in 2020, COVID notwithstanding, getting into the clinic and all the milestones that have been hit. But we're even more excited about what's to come, in particular for morf 057 in the early part of next year. So with that, maybe I'd turn it back over to Michael. Thanks very much for having us, and we're happy to take any questions from yourself or the audience if there are.
Yeah, fantastic. Well, thanks for that overview. You guys tag team that very nicely. A couple of questions. One is with the lead 057, Bruce, you gave a nice description of a lot of great preclinical data, a lot of proof of concept there, and then you described the phase 1, and you kind of listed 3 different parts: SAD, food effects, MAD, I think was probably the third part. While you would like to present it at a medical meeting, is there something there that would, drive the announcement of data? You don't probably want to see the phase 1 MAD portions. What do you need to see there to disclose it? And what would be meaningful for investors? Is that receptor occupancy? I mean, you said 100%. I don't know. And you want to see T cell data, right? T cells going up in the periphery.
Yeah, so maybe Mark could start there, and then I'll correct you on the 100%. I said we see 100% in the non-clinical models. I said that we're targeting over 90 in the human for sure.
Okay.
Excellent. I mean, I think that's a great question. There's a couple of questions in there. Let me try to unpack them. So I think for starters, just because of the staggered nature of the data coming into the management team, coming into the company with respect to the SAD first and then the MAD, there will be multiple months, potentially, uncomfortable months of sitting on such material data in advance of being able to present the entirety of the data at an actual conference by mid-year. And so as a result, in particular, because it still contains the receptor occupancy portion, right? So you asked, what do investors care about? What's relevant? Even in the SAD study, even though these are healthy volunteers, they still express alpha-4 beta-7. And even though it's a single dose, we can anticipate BID dosing and determine the trough levels of receptor occupancy by just measuring them 12 hours post-dose. So even with the SAD component of the data, we'll be able to get a tremendous amount of very relevant data, we feel, with multiple doses, by saying, at these doses, there's a safety profile, obviously, which of course is very important and part of any phase 1 trial. Safety, of course, but also PK. How much of the drug is getting into the blood at each dose? But then very importantly, the pharmacodynamic markers by dose to understand what percentage of alpha-4 beta-7 is being occupied in these folks 12 hours after dosing. And that is all available even in the SAD. Of course, the MAD will put a sort of exclamation point on those data, but even in the SAD, there's a lot of very relevant data there.
Right, so 12 hours, and that would be, to be fair, slightly conservative, because when you're doing cumulative dosing, you should get an accumulation of drug, like Bruce and Mark. So that would be the lowest. So even after 12 hours of a single dose, to me would be like, that's the lowest possible drug levels you could see with one dose. You would expect to see possibly good occupancy levels?
In a word, I think we'd say that yes, we hope to, and we want to see that dose-dependent differences as well. And we've been saying all along, long before we entered the clinic, that we hope to and plan to see 90+% occupancy at our relevant doses. So it's not impossible is the best way to answer that.
Hey, Michael, let me add just a little bit more to that. So when we look at the non-human primate experiments, and I'm only showing you the one graphic in the slides that we have here today, but it's actually fully discussed in the DDW poster that we gave this summer. If you look at the earlier time points of some of this, there's a little bit of variability in the early time points of the trafficking data, so the downstream PD effect. But when it comes to receptor occupancy, we're able to measure that very quickly. And it actually tracks very nicely based on the data we disclosed to DDW. It tracks very nicely with PK in non-human primates. So that's what we're looking for in human to see, does that PK to PD translate? And then in our MAD phase of these studies, we're going to also look at the other pieces that you brought up. We're going to look at T-cell trafficking. We're going to look at CCR9. We're going to look at all of the possible markers that we might be able to have our disposal of as well. The absent part of that story is that Vito has never been published for some of those T-cell trafficking, but we're going to look at it. We're relying on receptor occupancy because the key thing we want to get from this phase 1 study is safety, first and foremost, tolerability, and how high of PK and receptor occupancy can we get to in this study to be able to set our appropriate doses for our phase 2 study.
Because think about that, right? With PK, PD, and with your PD marker being receptor occupancy, right, then you should be able to go up, and you've probably modeled out, assuming it's well tolerated, you should have plenty of window to dose up and have coverage if you have a potent molecule.
Right. That's correct. And if you look at the doses that we're giving for our molecule in non-human primate, a 15 mg per kg oral dose is a very low dose of this. We saw the potency in the single-digit nanomolar region from our in vitro data, and we're able to really produce a nice sustainable response. The other thing I would mention about that is something you actually started to allude to with your hand signals, actually, Michael, and that is this is actually C trough, right? So this is low drug concentrations, not the high drug concentrations. So we also have most of the dosing period covered at a very high level of receptor occupancy. So even though 90% or greater is our target in human, we believe that for much of this dosing period will be at a very high level receptor occupancy.
Right. Let me be clear. Yeah. You're dosing up you'll be covering the target you're measuring down here. And I don't know how to think about the PK/PD effects of what the T cells are doing related to that effect. You see what I'm saying? It's different in oncology where you're just hitting and shutting down a target. Everybody knows the longer you've shut it down, that's good. I need to think about what that effect means to signaling the T cells to stop engaging. So we could talk about that later. But just to be clear, you guys would, based on receptor occupancy first from the SAD, that'd be important data for an investor to disclose, just on the SAD part.
We think so, yeah.
Okay, last question for you. I know because you teased me, alpha V beta 1 and alpha V beta 8 fibrotic. How far away is that from an IND?
So if we're able to get to a development candidate next year, we see an IND being 12 or so months behind that. So probably a 2022 IND. And then alpha V beta 8 is slightly behind that.
Okay, got it. Very good. Well, thank you guys very much for the review. Eagerly looking forward. You're underway in your phase 1. So it's not that far around the corner. And thank you for the update. We're very excited for you guys. So congrats and look forward to catching up.
Thanks very much. Appreciate it.
Thank you, guys.
Bye-bye.